Every eye doctor you've seen has been treating the wrong organ. Your vision is failing. And the cause isn't in your eyes, it's in your blood. 6 hidden mechanisms are shredding your sight from the inside, and almost nobody screens for a single one. Here's what nobody tells you. Long before anyone calls you diabetic, your blood sugar is quietly attacking the tiniest vessels in your body, and the ones feeding your retina are the thinnest of all. It doesn't take a high reading, just the swings, meal after meal, year after year, starving the vessels behind your eyes. Your night vision goes first, then colors dull, then the blur creeps in, and your eye doctor writes you a stronger prescription while the real damage keeps building where he never looks. 68-year-old woman. My patient. New glasses every 6 months, and her sight still sliding. Every eye exam came back clean, because the problem was never in her eyes. I ran her blood the right way and found it in minutes. We shut down the mechanisms one by one. 3 months later, her vision held steady for the first time in years. These 6 mechanisms are tearing at the vessels behind your eyes right now, and every one of them reverses once you know it's there. Cheap to fix, and no eye chart on earth will ever catch them. $46 billion a year, the eye care industry, selling you thicker lenses while the real cause blinds you from your bloodstream. Your fading sight is their paycheck. Now, number 2 on this list is the worst damage of all, and it's the one that hides the longest before it strikes. Every study is waiting for you in the description. Before we start, tell me one thing below. What's your age, and how fast is your vision slipping? I read every comment myself, and I write back. Now let's start with mechanism number 6, the hidden blood sugar trigger quietly destroying the vessels behind your eyes right now. Number 6, blood-retinal barrier breakdown. Your eyes have their own security system. It is called the blood-retinal barrier, and it works almost exactly like the blood-brain barrier you might have heard about. Think of it as a microscopic wall that lines the blood vessels inside your retina. This wall is made of tightly packed cells that control what gets in and what stays out. Nutrients pass through, oxygen passes through, but toxins, inflammatory molecules, and excess fluid are blocked. It is an incredibly precise filtering system. Now, here is the problem. When your blood sugar rises, even temporarily, it puts direct stress on these barrier cells. A 2019 study in Investigative Ophthalmology and Visual Science showed that blood sugar spikes as modest as 140 milligrams per deciliter, a level many people hit after a regular meal, caused measurable loosening of tight junction proteins in retinal barrier cells within just 72 hours in laboratory models. That means the wall starts to leak. And when it leaks, fluid seeps into the retina. Proteins that should never touch your photoreceptors suddenly flood the tissue. Inflammatory molecules pour in. This is what doctors call macular edema, and it is one of the leading causes of vision loss in people over 60. The worst part is that this barrier damage is cumulative. Every spike adds up. A 2020 analysis in the British Journal of Ophthalmology found that individuals with frequent post-meal glucose spikes above 160 milligrams per deciliter had a 42% higher rate of retinal fluid accumulation compared to those who kept spikes below 130 milligrams per deciliter. So what can you do about it? The most effective approach is to reduce the size and frequency of your blood sugar spikes after meals. Pairing carbohydrates with healthy fats, protein, or fiber slows glucose absorption dramatically. A study from Lund University found that simply eating vegetables before carbohydrates in a meal reduced the post-meal glucose spike by up to 35%. Apple cider vinegar diluted in water before meals has also shown a 20% reduction in post-meal spikes in a study published in the European Journal of Clinical Nutrition. Walking for even 10 minutes after eating has been shown to lower peak blood sugar by 22%, according to research in Diabetologia. These are simple, practical steps that directly protect your blood-retinal barrier from the leaking that leads to vision loss. The barrier can actually repair itself to some degree when you stop bombarding it with glucose spikes, but the older you get, the slower that repair becomes. Now, if that sounds concerning, the next mechanism is even more directly damaging to your retina. Number 5. Retinal insulin resistance. Most people think insulin resistance is a problem for your muscles, your liver, and your belly fat, but your retina has insulin receptors too. And when those receptors stop responding properly, things go wrong fast. Your retinal neurons, the cells that actually detect light and send signals to your brain, depend on insulin signaling to survive. Insulin does not just manage sugar in the retina. It activates survival pathways that keep neurons alive and functioning. When insulin resistance develops in the retina, those survival signals go dark. A groundbreaking 2017 study from Penn State College of Medicine found that retinal insulin resistance developed even before systemic insulin resistance was detectable in blood tests. In their animal models, retinal neurons began dying 6 weeks before any other organ showed signs of insulin resistance. That finding is stunning because it means your eyes could be the very first organ damaged by insulin problems, long before you ever get a warning from a standard blood test. The researchers measured a 38% decrease in retinal neuron signaling within the first 8 weeks of high sugar feeding in their models. Another study published in Progress in Retinal and Eye Research in 2020 found that restoring insulin sensitivity in retinal tissue reversed early neuronal damage by up to 45%. The neurons were not dead yet. They were starving. They were losing their survival signals. And when those signals came back, the cells recovered. So how do you restore retinal insulin sensitivity? The most evidence-backed approaches are regular physical activity and reducing refined carbohydrate intake. A 2018 study in diabetes care showed that 150 minutes per week of moderate exercise improved whole-body insulin sensitivity by 25%, with corresponding improvements in microvascular function, including the tiny vessels in the eyes. Intermittent fasting protocols have also shown promise. A 2019 study in Cell Metabolism found that time-restricted eating within an 8 to 10-hour window improved insulin receptor sensitivity by 19% in just 5 weeks. Magnesium supplementation is another powerful tool. A meta-analysis of 18 studies published in Nutrients found that magnesium intake of 350 milligrams per day improved insulin sensitivity by 17%. Foods like pumpkin seeds, dark chocolate, spinach, and almonds are among the richest sources. The key takeaway here is that your retinal neurons are not just passive recipients of blood sugar damage. They have their own insulin system, and when that system breaks, they begin to die. But the next mechanism takes this cellular damage to a completely different level. If you're serious about staying healthy, active, and independent after 60, hit like and subscribe now. The next insight may be the most valuable one we share today. Number 4. Oxidative stress overload. Here is something most people do not realize. When your cells process glucose for energy, they produce waste products. Think of it like exhaust from a car engine. Under normal conditions, your body has cleanup crews called antioxidants that neutralize this waste. But when blood sugar is elevated, glucose metabolism goes into overdrive. The exhaust production skyrockets, and your antioxidant defenses simply cannot keep up. This imbalance between waste production and cleanup capacity is called oxidative stress, and your retina is uniquely vulnerable to it. The retina consumes more oxygen per gram of tissue than any other organ in your body. It is the single most metabolically active tissue you have. That means it produces more oxidative waste than anywhere else. And when blood sugar pushes that production even higher, the damage is devastating. A 2018 study published in Free Radical Biology and Medicine measured oxidative stress markers in the retinal tissue of individuals with elevated blood sugar. They found that reactive oxygen species, which are the specific molecules that cause oxidative damage, were elevated by 63% in people with fasting glucose above 110 milligrams/dl compared to those below 95 milligrams/dl. That 63% increase translates directly into accelerated destruction of photoreceptors, the cells responsible for your ability to see detail and color. Another study in antioxidants and redox signaling from 2020 showed that retinal cells exposed to high glucose conditions had a 47% reduction in their natural antioxidant enzyme activity, specifically superoxide dismutase and glutathione peroxidase. So not only does oxidative waste increase, but the body's ability to clean it up simultaneously decreases. It is a double hit. The good news is that specific nutrients can dramatically shore up your retinal antioxidant defenses. The AREDS2 study, one of the largest eye nutrition studies ever conducted with over 4,000 participants, found that a combination of lutein, zeaxanthin, vitamin C, vitamin E, and zinc reduced the risk of advanced vision loss by 25%. Lutein and zeaxanthin are particularly important because they concentrate specifically in the macula, the part of your retina responsible for sharp central vision. Egg yolks, kale, spinach, and orange peppers are among the best food sources. Astaxanthin, a powerful carotenoid antioxidant found in wild salmon and krill, showed a 54% reduction in retinal oxidative markers in a 2019 study published in Marine Drugs. Even 6 milligrams per day showed significant protective effects within 8 weeks. And as damaging as oxidative stress is, the next mechanism adds fuel to an already burning fire. Number 3. Chronic microinflammation. Inflammation is your body's natural response to injury or infection. In short bursts, it is helpful. It brings immune cells to a problem area, cleans up damage, and starts repair. But when blood sugar stays elevated or spikes repeatedly throughout the day, it triggers a very different kind of inflammation. This is low-grade, chronic, systemic inflammation. It does not make you feel sick. There is no fever. There is no swelling you can see. But inside your retinal blood vessels, it is causing constant, relentless damage. Elevated blood sugar activates a molecular pathway called NF-κB, which is essentially the master switch for inflammatory gene expression. When NF-κB is activated, your cells start pumping out inflammatory molecules called cytokines, specifically TNF-alpha, IL-6, and IL-1 beta flood the retinal tissue. A 2020 study published in the Journal of Neuroinflammation measured cytokine levels in the vitreous fluid, that is the gel inside your eyeball, of patients with early retinal damage. They found that TNF-alpha levels were 3.2 times higher and IL-6 levels were 4.1 times higher compared to healthy controls. These inflammatory molecules do not just float around harmlessly. They actively destroy the parasites, which are the support cells that wrap around your retinal capillaries and keep them structurally sound. When parasites die, capillaries become weak, leaky, and eventually collapse entirely. A major 2019 study in diabetes found that chronic low-grade inflammation accounted for approximately 40% of parasite loss in retinal tissue samples. That parasite loss is considered one of the earliest and most critical steps in diabetic retinopathy, and again, it happens even at pre-diabetic sugar levels. Fighting this chronic inflammation requires a multi-angle approach. Omega-3 fatty acids are among the most potent natural anti-inflammatory compounds for the eyes. A 2021 meta-analysis in the American Journal of Clinical Nutrition covering 12 studies and over 31,000 participants found that those with the highest omega-3 intake had a 29% lower risk of retinopathy. Wild-caught fatty fish like salmon, sardines, and mackerel eaten 3 times per week provide therapeutic doses. Curcumin, the active compound in turmeric, has shown remarkable anti-inflammatory effects, specifically in retinal tissue. A 2019 study in molecular vision found that curcumin supplementation reduced retinal NF-κB activation by 52% in just 6 weeks. Taking it with black pepper increases absorption by 2,000% according to research in Planta Medica. What comes next, though, is a mechanism so sneaky that most doctors do not even check for it. Number 2. The sorbitol trap. Inside your cells, there is a metabolic pathway that most people have never heard of. It is called the polyol pathway, and under normal conditions, it is barely active. It processes less than 3% of the glucose your cells take in. But when blood sugar is elevated, something shifts. The main pathway for glucose processing gets overwhelmed, and the excess glucose gets shunted into the polyol pathway. Here, an enzyme called aldose reductase converts that extra glucose into a sugar alcohol called sorbitol, and this is where the trouble begins. Sorbitol cannot easily pass through cell membranes. It gets trapped inside. As it accumulates, it creates massive osmotic pressure, essentially sucking water into the cell like a sponge. The cell begins to swell. In the lens of your eye, this swelling distorts the carefully organized protein fibers that keep your lens crystal clear. The result is clouding, which is exactly what a cataract is. A 2018 study in experimental eye research found that even moderately elevated blood sugar, fasting levels between 100 and 125 milligrams/del, increased sorbitol accumulation in lens cells by 280% compared to levels below 90 milligrams/del. That 280% increase was directly correlated with lens opacity changes visible on examination. But sorbitol does not just damage your lens. In your retina, sorbitol accumulation depletes a critical protective molecule called myo-inositol. Myo-inositol is essential for nerve signal transmission in your retinal neurons. Without it, signals degrade, visual processing slows, fine detail becomes harder to resolve. A study in the Journal of Diabetes and its complications from 2019 measured a 34% reduction in myo-inositol levels in retinal nerve tissue in the presence of elevated sorbitol. Participants in that same study showed measurably slower visual processing speeds. To combat the sorbitol pathway, keeping blood sugar below 100 milligrams/del fasting is the most direct strategy, but specific nutrients also help. Alpha-lipoic acid has been shown to inhibit aldose reductase activity by up to 40% in a 2017 study published in Diabetic Medicine. A dose of 600 milligrams daily showed significant results within 12 weeks. Quercetin, a flavonoid found in onions, apples, and berries, also inhibits aldose reductase. Research published in the Journal of Agricultural and Food Chemistry showed a 37% reduction in enzyme activity with regular quercetin intake. Maintaining adequate B1 thiamine levels is also crucial. A 2020 study in Diabetologia found that high-dose thiamine, 300 milligrams per day, reduced sorbitol accumulation in tissues by 25% within 8 weeks. Benfotiamine, a fat-soluble form of B1, showed even stronger results at 32% reduction. And now we arrive at the number one mechanism. This is the one that changes everything. Number 1. Advanced glycation end products. This is the big one. Advanced glycation end products, known as AGEs, are perhaps the single most destructive force acting on your retina after 60. And here is the terrifying part. Once they form, they are essentially permanent. AGEs are created through a process called glycation. When blood sugar is elevated, glucose molecules attach themselves to proteins and fats in your body without any enzyme controlling the process. It is a random, chaotic chemical reaction. Once glucose bonds to a protein, it undergoes a series of chemical rearrangements over days and weeks until it forms a hardened, cross-linked structure. That structure is an AGE, and it never lets go. These AGEs accumulate in tissues throughout your body, but they concentrate heavily in the retina due to its high metabolic activity and abundant blood supply. A landmark 2020 study published in Diabetes Care measured AGE levels in the retinal tissue of over 1,500 participants aged 60 to 85. They found that individuals in the highest quartile of AGE accumulation had a 370% higher risk of significant retinal damage compared to those in the lowest quartile. That is not a typo. 370%. The mechanism of damage is multifaceted. AGEs directly stiffen blood vessel walls, reducing their ability to expand and contract. They activate a receptor called RAGE on the surface of retinal cells, which triggers cascading inflammatory and oxidative responses. A 2019 study in the Proceedings of the National Academy of Sciences found that age-range activation increased retinal cell death by 58% and boosted inflammatory cytokine production by 240%. Ages also cross-link collagen in the vitreous humor, the gel inside your eye, causing it to shrink and pull away from the retina. This traction is a major cause of retinal tears and detachments in older adults. A 2021 study in ophthalmology found that age-related collagen cross-linking in the vitreous was 3.5 times more advanced in people with elevated long-term blood sugar averages. Reducing age formation and accumulation is absolutely critical for protecting your vision. The most important step is keeping your average blood sugar as low and stable as possible. Every point reduction in HbA1c, the 3-month blood sugar average, reduces age formation proportionally. Cooking methods matter enormously too. High-heat cooking like grilling, frying, and broiling creates dietary ages in food. A 2017 study in the Journal of the American Dietetic Association found that switching from high-heat to low-heat cooking methods like steaming, poaching, and stewing reduced dietary age intake by 50% and measurably lowered circulating age levels within just 4 weeks. Carnosine, an amino acid found in meat, has shown the ability to prevent glycation. A 2019 study in amino acids found that 1,000 milligrams daily of carnosine reduced age formation markers by 28%. Benfotiamine, which we mentioned earlier, also blocks age formation through a separate pathway, reducing it by 40% in a 2018 study in Diabetes Technology and Therapeutics. Finally, the spice cinnamon has shown remarkable anti-glycation properties. Research in the Journal of Medicinal Food found that just half a teaspoon of Ceylon cinnamon daily reduced glycation markers by 21% over 12 weeks. And that brings us to the end. These 6 mechanisms are not separate, isolated problems. They feed into each other. Oxidative stress worsens inflammation. Inflammation accelerates age formation. Ages increase oxidative stress. Sorbitol accumulation weakens barriers. Insulin resistance amplifies everything. It is a vicious cycle, and blood sugar is at the center of every single loop. The good news is that you now know exactly what is happening and exactly what to do about it. You do not need expensive treatments or complicated protocols. You need stable blood sugar, the right nutrients, smart meal timing, and consistent movement. These are simple, accessible changes that can dramatically slow or even partially reverse the retinal damage these mechanisms cause. Your eyes are not failing because they are old. They are failing because of what your blood sugar has been doing to them for years. And now that you understand that, you have the power to change it. If this information was valuable to you, please hit that subscribe button so you do not miss our next video. We break down health topics like this every week with real science and real solutions. Thank you so much for watching.